disorders seemed very tractable. And I just got that recording thing, Jim. Okay.
Okay, I apologize. I should have done that earlier. I'm sorry.
No, that's okay. I just okayed it, so we're all good. Okay. So I'm gonna see if I can. So in around 2017, this was our entrance into the IBM field. We published a paper, and uh, just getting back to Suds and I were talking before the call about Gary Beyer, who was an IBM patient in Wisconsin. And Gary was one of the first people to contact me after this paper was published. This was a paper looking at TDP43, this gene that I'll talk about extensively, and how we can model this gene in skeletal muscle. And it was shortly after we published this paper that I started to get contacted by patients with IBM because people felt like this would be our entranceway to new therapies. Okay, so if anybody wants this paper, I'm happy to send it to Jim, for example, and you could forward it on to whoever might want to see this.
Of course.
And so around that time, this is now 2018, here's a picture of Gary Beyer and his wife, Julie. Um, we had extensive ongoing conversations about IBM. I learned a lot from him. I learned about his struggles. I learned about what muscles were weak. I learned about what he tried. And he was such an advocate for the disease, and very unfortunate for me to just hear that he passed away. But he was one of the first people that I connected with on the patient side of things that really clued me into the struggles of IBM and how devastating it can be. And Gary wrote this article in his local newspaper, the Oshkosh Herald. This is a local Wisconsin newspaper. And he talked about work that we're doing. And he even went so far as to set up a GoFundMe account to raise money to support research in my lab. Keep in mind, I had not known Gary, but my impression is that he was this kind of a person, selfless and driven to bring people together and do real good for this disease. And so we started to raise a little money, and we started to get new things off the ground and connect with patients. And so that's why I think this type of a call is so important, so that I get to meet the people that are actually affected by this disease. And so this got me very interested in IBM because, unlike a brain or a spinal cord, the muscle is accessible. And in fact, you can potentially devise therapies that go directly into the muscle via intramuscular injections. One of the problems we had was coming up with ways to get therapies into the brain through the blood-brain barrier, which is a very difficult thing to penetrate. Things don't always want to get into the brain, whereas the muscle is much more penetrable. So I thought, gosh, could we make some headway in IBM? So this was our entrance to IBM, and it really came late in the game. This was after I had already established my lab. But sometimes you need people to really clue you in and get you excited about things, and I think Gary was that person. So this was a letter I hope you all have. When Jim reached out to me, he had asked if I could advocate for veterans' health benefits because IBM was not considered a disorder that would allow you to have those benefits. And I thought, how could this be? We always viewed IBM as ALS of the muscle. And so I wrote this letter, and it sounds like you all have it, but feel free to distribute it. And I hope that it allows you to achieve those benefits, because I really do feel that way about this disease. In this letter, we refer to IBM as ALS of the muscle. There's many overlaps between IBM and ALS. In fact, we view these two disorders along a similar continuum. One targets TDP43, this protein that is devastating to the muscle, and the other targets it to the brain and the spinal cord. And wherever it emerges is likely the tissue that's affected. And so we do view them in that way. Military veterans represent a population at high risk. Why that is, we don't know. I'm happy to talk about it. Veterans with IBM should be granted much deserved benefits due to this overlap. Okay, so here's what my lab studies, essentially these four things. Why do some of us get IBM and related disorders and others don't? We would call these risk factors. So that's number one, is the risk factors that predispose you to get disease. How do your genes determine whether or not you would get IBM? We would refer to this as your genetics. Everyone has different genetics. Some people's genetics might predispose them a bit. And when you combine that with other risk factors, all of a sudden you might tip over the edge. Okay. What is it about aging that's a risk factor? Sometimes we don't think about this. Aging is one of the most important risk factors in all of these diseases. We call them age-related degenerative diseases. Children, young children, tend to not get these diseases. So there's something about aging that predisposes us. So we're very interested in what it is about time that can really act as a dominant risk. And then, of course, this last one is what we all want, right? Can we develop stuff? And I say stuff because typically we think of drugs, pills, right? But guess what? It doesn't always have to be a pill. Sometimes it can be an immunotherapy. Sometimes it can be a virus. And so can we develop stuff? What the stuff is, we don't so much care as long as it achieves the desired end goal. And of course, we want number four. We all want number four, right? But to get to number four, we need to understand number one, number two, maybe number three, but definitely number one and number two. So it's hard to jump to four. And I think in the past, we've tried to jump to number four. So what I would say is if we go through the motions in the right disciplined manner, we can achieve number four, but we can't jump to it. We have to do it stepwise, okay? Here's how complicated this disease is for anyone that doesn't that doesn't have a background in molecular and genetics, which I'll assume that you don't for the time being. But if you have any questions about this, I'm happy to answer that. But I just want to illustrate how complicated this is. Here's a family, and this is a pedigree chart with a husband and a wife. The males are in the squares and the females are in the circles. And here's all their children, okay? And here's another, this is another family. This is a wife and a husband. They had a son. This son ended up having a mutation, a genetic mutation that led to frontotemporal dementia, okay? This son from this family married this daughter from this family. They had 1, 2, 3, 4 kids. 2 of their kids had this mutation, this exact same mutation that their father had. But the son had IBM, and this son had ALS. So think about this. This is a single mutation, the same exact spot on the chromosome. Dad has FTD, son has IBM, and son has ALS. So it's actually hard to believe, but it just illustrates, and this is a picture of one of the sons. This is a son that had the myopathy, and you can see there's some deltoid atrophy, and so this is a verified myopathy patient. And so what this illustrates is that there is massive, we would call this heterogeneity. In other words, you can plop the same exact genetic mutation into a series of people, and it can manifest itself in totally different ways. So really, really bizarre, but really informative. It tells you that there are other factors that drive a certain disease. It also tells you that these diseases are intertwined. They're not one disease. They're a continuous spectrum. And in fact, ALS patients can have FTD, and FTD patients can have ALS. And now they're slowly working in IBM into this equation to figure out where IBM lies on this spectrum. Not quite there yet, but I think that's emerging. And so this is how you look at IBM when you take a tissue biopsy. This is a muscle biopsy. And so you can see this is an example. These are nice, round, large muscle fibers. And then occasionally you get a regenerative muscle fiber that has all these, that's smaller and also looks like it's trying to regenerate by putting its nuclei in the middle of this fiber. And so these are all different stains that you can do using biopsied muscle tissue. These are rimmed vacuoles. These are these large structures in white. Those are another hallmark of IBM. And so with IBM, you can diagnose it. You can be told you have a myopathy. What we can't do is treat it very well, which I'll get to in a second. But anyone with IBM has this characteristic inflammation in the muscle and myofibrillar degeneration. Those are the two hallmarks of IBM. And people debate about which one to go after. Should we use anti-inflammatories or should we try to rebuild the muscle by getting rid of what's causing this? There are two camps that tend to debate each other on that one. Happy to talk about that at the end as well. In 2006, the protein that is thought to cause the degeneration, certainly in ALS and frontotemporal dementia, was this protein called TDP43. Now, I had the good fortune of being in this lab at the time that this was discovered. This was in 2006 when I was at University of Pennsylvania. And so when this paper was published, it was game changer is an understatement. This was the most remarkable discovery because this protein ended up being important, not just in ALS. This is an ALS patient spinal motor neuron. This is the whole cell. You're seeing the outline of it. And this giant clump in the middle is the trash, the aggregates, the junk that accumulates with age and over time that ends up killing these neurons. This is the stuff that we want to get rid of. This ends up being the same stuff that's in frontotemporal dementia patients, the majority of them, and also in IBM patients. And we think that this might be the stuff that's causing the degeneration and muscle weakness. And actually, I believe that that's shown here. So here's the same motor neuron in an ALS patient. If you take this junk and you put it into the brain instead of the spinal motor neuron, it ends up forming in your neurons in your frontal cortex. In that case, you get FTD. If this junk ends up forming in a skeletal muscle, you end up getting what looks like a myopathy. In this case, this is a SIBM biopsy. And you can see that all this stuff in orange and red here is the TDP43. So we have one protein that we can attack. So to me, that's a great thing. That means if we get rid of it, we could potentially have a treatment for not just IBM. Of course, we want to treat IBM, but also it could double over or triple over as a treatment for these other conditions. Okay, so that was the idea. So TDP43 is this really amazing protein. It ends up accumulating. So it ends up accumulating as aggregates in the brain, in the spinal cord, in the muscle. In ALS patients, for a long time, people thought it only accumulated in the brain or the spinal cord. But it turns out more recent papers are showing that even in ALS patients, the TDP43 accumulates in muscle. So these are muscle biopsies from 30 different ALS patients. And in every single one, they showed hallmarks that look like IBM. So think about this for a second. This is an ALS patient. These are not IBM patients. These are images that show that in ALS, these patients have what looks like IBM in their muscles. It's also in their spinal cord and brain, but it's also in their muscle. Okay, so this is illustrating that there's this continuous spectrum again. It's not always going to be a clinical, you have this, you have this, you have this. It's looking more like you have this gamut of syndromes that all are sort of coalescing. Okay, so this is a very recent study, so 2019. So we're learning about this over the last few years. Here's another really remarkable study, and I'm happy to send you these slides, by the way, Jim, if anybody wants to have these references.
That would be very helpful. Thank you.
Absolutely. Here's a paper that just came out. This is a paper showing that if you have a mutation in TDP43, here's the TDP43 gene down here. If you lop off about 10 amino acids in the protein, these patients that are shown in this paper, they don't have ALS, they don't have FTD, they have inclusion body myositis. So you can have a mutation. You can have a mutation in TDP43 that doesn't cause the other two disorders, but that causes IBM. And so, these are all very striking. And for me, these are all coming out in real time. This is all new to everyone. So we have to take this and try to understand what it means so that we can treat patients. Okay. And so this is a paper that just came out this year. Here's an example. This is the TDP43 in an IBM patient, and this is the TDP43 that's in a normal patient. All this green stuff that's in these muscles is the extra aggregated, you can see it here, the extra aggregated TDP43 that is causing these muscles to deteriorate. And it's mutant. It's this genetic mutant. And you can see from this family how extensive it is. Here's mom. She gave it to all of her kids, and etc., and on and on and on, and it propagated down. And this is what, and inevitably, many of these children and their children's children end up having this phenotype. So TDP43, when mutated, can cause inclusion body myositis. A very, very important point.
Dr. Cohen, if I could interrupt here, that is a fascinating illustration there. But would that be considered hereditary IBM or a familial IBM as opposed to sporadic IBM?
That's right. This would be considered the familial, a familial IBM. And so I'm kind of grouping them. The only difference to me is that familial IBM can be traced back within a family, within a kindred, they would call it, whereas sporadic IBM could be due to, it's just an isolated incident that there's no family history of. Now, they still could have similar etiologies of their disease, but one, the family history, and the other, you don't, if that makes sense, Jim.
Yes, indeed. Thank you.
Oh, okay. So here's what, if this was too sciencey, these are the 4 takeaways from this. 1, myopathies, including sporadic IBM, present differently in different people. You can take the same mutation and put it in a different person, and they get a totally different clinical looking disease. They can have overlapping symptoms. In other words, a single patient can have ALS, dementia, a particular disorder of the bone, and IBM, and they might be merged. They might be co-symptomatic. So that's another important point. You can have multiple symptoms within the spectrum. These TDP43 aggregates in the muscle are likely a causative factor. And so what this means, and I say likely because we're still in this, investigation, curiosity phase, but in my opinion, they're likely a driving force. And so we need to figure out how to get rid of them. Why they form is not known, okay? All right, so if you go to the Myositis Association homepage, they say the underlying causes of many of these myopathies are not known, but some people believe that there is an environmental exposure connection that triggers disease. And let me just move my, sorry, give me 1 second here. Okay. And so some of the environmental risk factors that have been implicated, but there's not much that's solidly supporting these. And I just want to, I know that that this audience may want to know things like, what are the factors that might promote IBM? Is there evidence linking these? And I wish that we had the answer. It's a very difficult question to answer, but there have been implications, stories, there have been reports that viruses, bacteria, UV radiation, smoking, exposure to drugs, biologics, and supplements could potentially cause inflammation, for example, and lead to a risk to IBM. But you have to be very cautious when you say that, because people move, people get off medication and start new medication. So there are so many variables here that it's been very difficult to hone in on the epidemiology of whatever the factor that you might be interested in. But there is definitely a thought out there that there's an environmental link, okay? And so the idea is that your environmental risk factors, whatever they may be, we sort of favor the idea that there are environmental chemicals that we're breathing in that may put you at higher risk. That's something we're currently working on. But all of these things could be grouped into environmental risk factors. When you combine those with your personal genetics, you create a human, a human body that might have an elevated risk to develop this spectrum of disorders. This would include IBM. And so we think that there's a bucket, and when that bucket is full, it might put someone over the edge. And so your genetics, you may not be able to do much about. But if these risk factors end up being important, then there are strategies for intervention here or mitigation. You avoid being exposed to these things, lifestyle interventions, that type of thing. This is all a work in progress. I wish I knew more, and I think things we're doing in the lab are going to flush this out a little bit more, okay? So there was this really interesting study done on ALS patients among veterans. And what they found was that essentially military veterans were at twofold higher risk of ALS. And when I saw that, of course, immediately I thought, well, they must be at higher risk for other diseases as well, because if ALS lies in a spectrum, you might expect them to be at higher risk for other things as well. And actually, that's true. They're at higher risk for Alzheimer's disease. They're at higher risk for Parkinson's disease, at higher risk for frontotemporal dementia. IBM, a little bit less studies to support that, but certainly the others are there. And the other interesting thing was that there was highest prevalence among those in the Air Force. And so we're not quite sure what that means, but it could mean that there's a connection between exposure, or that there's a connection between geographic location, or that there's a connection between whatever physical trauma might be exerted on certain populations in the military. But there's not a lot on this. This is sort of early days. And I think it's complicated by other things, your other lifestyle factors, where you've lived, that sort of thing. So we really need good epidemiology to support this. And so that's what we're trying to do in collaboration with epidemiologists at the moment.
Dr. Cohen, that was a very interesting revelation here, is that I will explain to you that many years ago, several of us from the Chicago area had lunch together. Ed Arnold, who's here today, myself, and actually Dave Mokel, who is now the chairman of the board of the Myositis Association, along with another doctor who we were all in the same support group. During that lunch, it was a very casual, sort of a spur of the moment lunch. We found out that 3 out of the 4 of us, and we all had IBM, and 3 out of those 4 were Air Force veterans. And I will tell you that that is what triggered my digging into this from the military perspective. And now that you bring this up, statistically, it is really really eye-opening. The one thing that I would offer to you that may be different than other branches of the service is that, unlike the other branches of the service, all Air Force personnel go through basic training at the same place, which is Lackland Air Force Base in San Antonio, Texas. And that is a common thread. And when you go down there, you run through a gauntlet of medical technicians that are injecting you with God only knows what, but it is the common thread that every Air Force veteran goes through that same location and goes through that same process, whereas Army, Navy, Air Force, Army, Navy, Marines, Coast Guard, they are separated for their basic trainings, where Air Force is concentrated in one place.
And where is that, Jim? In Texas?
In San Antonio, Texas. It's in all of our basic training was done at Lackland Air Force Base in San Antonio.
Got it. Yeah, and I think that's one of the advantages of these types of calls is you can sit down with people and sort of dig through your past and see what are the things that we might have in common that someone like me could then go and test in the lab. So based on what you said, I'm going to look into what happens there and what the testing might be like. I think that's really interesting, unless there's something else that Air Force personnel do that's different. That seems like one to follow up on. Thank you, Jim.
Thank you.
Okay. The other thing that is worth mentioning is you can get ALS purely from exposure to an environmental toxin. If any of you have heard of this disease called ALS of Guam, this is something I learned about once I set up my lab and started digging into ALS. This is a patient who feasted on raw bats. In Guam, bats are a delicacy. And through 20 plus years of detective work, it was shown that a toxin that was present in the bats, which was leached into these nuts, these are cicad nuts that are in Guam, these nuts harbored a toxin, and the bats would eat the nuts at night, and then the humans would eat the bats. Now, had they cooked the bats, this wouldn't have happened, but they were eating these bats raw as a delicacy. And so ALS of Guam, which was essentially ALS, was happening at 100-fold higher incidence than normal. It was shocking. The investigator who discovered this, Paul Cox, said to these folks, listen, stop eating raw bats. If you stop eating them, you won't get ALS. Sure enough, they stopped eating these bats, and the incidence of ALS on Guam went away. What's the lesson here? The lesson is that you can get ALS purely from exposure to an environmental toxin. Now, ALS is not IBM, but I say this because ALS is on this continuum, this spectrum. So if this was to hold up for other disorders, the implications are that IBM could be a disorder that is responsive to environmental triggers. And so this was really sort of the ultimate in detective work. And if anyone is interested in reading more about this story, just search Chamorro people of Guam. And it is really a eye-opening story about how following a herd mentality, sometimes built into your culture, can really lead to some devastating consequences, at least medically. Okay, and this is a picture of the US. This is always a little surprising to me, but ALS in the US is not entirely evenly distributed. So look at the left half of the US compared to the right half of the US. We see higher incidences here in the South, in the Northeast, and the incidences are much lower out West and Midwest. So this is a little surprising. You might expect incidence of ALS. Now, we don't have a comparable map for IBM, or at least I'm not aware of one. If anyone does, I'd love to look at it. But at least for ALS, we see a non-even distribution, which again could be linked to exposures in certain parts of the country, all supporting this environmental link, if that makes sense. So what are we doing about this in a lab? Well, first of all, here is a very talented investigator in our lab. Her name is Julia. Julia said to me one day, I would love to screen all of the things in the environment that could cause IBM, ALS, FTD. Why don't we take all the thousands of pollutants, toxicants, exhausts, fuel exhausts, pesticides, things we're spraying on crops? Why don't we take them all, put them in one of these giant multi-well plates that can hold thousands of chemicals, and test each one for its ability to cause TDP-43 to start to aggregate. And so this is the kind of study that's ongoing. It's happening right now. And I can tell you, out of thousands of chemicals, we found 7 that we think are actual mediators that drive our TDP-43 to want to aggregate. And so maybe next time I talk to you, we'll have more insight into this. But this is happening at the moment. And we're really excited because we think we found a few that are very common that might be sprayed on crops, that might be out in the agricultural. And if they are, then it's possible that either farmers or other agricultural workers or even the military are at higher risk once they're out and about. And so this is all ongoing, but I hope to have more to tell you about this next time I talk, and I'll certainly keep you updated on this. All right, can we design more effective drugs for IBM patients? Well, what's out there right now, I wish there was more. There's really not a whole lot. There's lots of clinical trials. There were some that failed. So there was a trial for a drug called arimoclomol. Some of you may or may not be familiar with this drug. This is a drug that's supposed to increase your resilience by inducing chaperones that are supposed to refold all your aggregated proteins. This was tested in phase 2 and phase 3 way back in 2016, and it hasn't really met criteria for a successful endpoint. So this had promise, but this particular drug wasn't as effective. There are other drugs that are currently ongoing that might act as chaperone inducers, but nothing has really looks promising at the moment. So we took a chemical called HSF1A. This is a drug that we got from a paper that was published here. This is a paper from a person named Dennis Thiel, who happens to be at Duke University. And Dennis said to me, why don't you test our compound, this HSF1 activator? This is a drug that's thought to activate this protein that might refold all of your TDP-43. And sure enough, when we put this into cells, you see all this green, all these green blobs? This is all aggregated TDP-43. Look what happens when we added this drug. A lot of it, not all of it, a lot of it went away. And this is the graph to show that. If you have 100% IBM type muscle, then when you treat with this HSF1 activator, look, it goes down to 40. So I also want you to realize we don't need to get rid of your TDP-43. It could be that we partially restore muscle function by just preventing TDP from aggregating just a little bit. So I don't want this to be an all or none. Right now, IBM has no real effective treatments. So if we can just limit your TDP-43 from accumulating in your muscles, you'd feel a whole lot better. So this is a drug we want to advance, and we're now testing this in mice and other animal models. So also stay tuned. We'll see what we find. We have other drugs. So here is a drug that prevents inflammation, okay? And I just told you that IBM is a disorder of inflammation as well. Your muscle cells, your fibers are loaded with inflammatory T cells, and they're thought to drive non-productive inflammatory reactions. So here's a drug that when we added back to, now these are neurons. We haven't tested this in muscle fibers yet, but we will. Here's a drug that when we added it to neurons, it made the neurons very healthy. So we added this drug and these neurons got really healthy. The way we tell health of neurons is when they look very spotty like this, they're not healthy. So as soon as we added this drug, these neurons got very healthy. And here's a drug that we would refer to as amazing. This is a drug that when added in the presence of inflammation, made these neurons amazingly healthy. So we have HSF1A, which might help boost your TDP, get rid of your TDP43. And now we have anti-inflammatory drugs that we haven't really characterized well that might limit inflammation. Both are on our agenda in terms of testing them in mice and eventually in people. Okay, so here was the study that we published that got a lot of excitement. And why is this study appropriate for the IBM community? I just want to show you a couple snippets of information that got us really excited. First of all, here's muscle fibers. This is a muscle fiber from a mouse that we injected with TDP43. Here's the TDP43 in green in this mouse. And when we did this, this mouse got an IBM-like phenotype. This mouse started to recruit T cells. These are T cells in red. These are the cells that set off this inflammatory response. And this muscle fiber started to deteriorate and die. Essentially, we recreated IBM by injecting TDP43 into a mouse muscle with a needle, and that was all it took. So this was really, really cool to us because now we have a model that we can test therapies, drugs, etc. And so when we compared these muscle fibers to what an actual patient looks like, so here's our mouse and here's our patient. They ended up looking very similar. This is a patient with a degenerating muscle fiber that has TDP43 and also a T cell response. And that's exactly what we got here. So now what we're going to do is we're going to try to cure these mice by treating them with drugs like HSF1A or these other anti-inflammatories. If that works, we hope to get this new era of drugs out into people. Of course, it'll take some time, but they'll work our way through from phase 1 up to phase 3 clinical trials. And that's the process, which could take anywhere from, up to 5 to 10 years, but it has to start somewhere. And so we hope we can develop and characterize these new drugs that will be helpful to you all at some point. Here is a muscle fiber loaded with TDP43. Look at this. All this stuff in green is this TDP43 that is damaging your muscle fibers. So we took another approach. We said, let's just deliver the gene to these mice. This gene called HSF1, it's not a drug, it's a gene. So we put it on a virus and we delivered it to these muscles. And look what happened. In the presence of this gene, all of this TDP43 that is the cause, we believe, of IBM is now gone, mostly gone. And so we feel like delivering the gene might be a really viable therapy. So how do we deliver this gene? It would have to be delivered on a virus. And so we think that another option for us to consider is to take a gene, package it in a virus, and deliver it to the muscles of patients with IBM. It certainly worked well in mice, and so we think it'll work well in people. Here's another example. Here's a muscle fiber that didn't get HSF1. I'm circling it right now. It's got all the TDP43 that's killing the muscle fiber. Here is a muscle fiber that got HSF1. And guess what? There's very little TDP43 in this muscle fiber. So we think the presence of HSF1, this gene that we're delivering, is sufficient to lower your TDP43. And we could quantify that here. So we always like to show graphs when we show science, because you need a number. You need to put a number on these things. Here's where we were in a full IBM model, and here's where the presence of our HSF1 took us, all the way down here. So we essentially completely rescued these IBM muscle fibers. This is what we'd love to get into people, this particular gene, and that's what we're trying to do. So what's the idea? Wouldn't it be great if we could take an IBM patient, package a gene, in this case HSF1, into this virus. This would be a beneficial virus. This wouldn't be a bad virus. This would be a good virus. Put this virus into people, maybe inject it intramuscular. And what we would expect, if everything we've done in animals is true, we would expect this healthy muscle I'm sorry, this is backwards. We would expect this inclusion body myositis muscle to go the other way and revert to a healthier muscle. Okay, so disregard this. This was a typo. This should have been going the other way. But we think this gene will revert the IBM muscle back to a healthy phenotype. And this is very doable. This is very doable. At the moment, there are lots of models of viral treatment. And in fact, people with spinal muscular atrophy is a disorder that I can think of that's currently being treated with helpful viruses. So I don't think this idea is out of the ordinary or bizarre. This is something we could do. We would test it in mice, and if it looked promising, we would test it for safety and tolerability in people. And if it looked promising, here's a drug that could be out on the market. We just have to go through the steps of testing it. So what do we need to do to go through these steps to test it? Well, here's our virus, which we currently have. This is a virus that we package. See these little particles? This expresses our gene called HSF1. We inject this virus into mice at first. And here's a cute little mouse. We inject this mouse with the virus, and then we test this mouse for its stamina, its muscle exertion, its weakness, its muscle force. We can do all of those things. This is one test I'm showing here, which is called a locomotor test or a rotorod test. And each of these mice has to run on the rotorod. And when they're weak, so this is a rotating rod. And when they're weak, they fall off. Okay, and you can tell when the mouse fell off because it hits this button. And so when we would have some mice that have IBM, they would fall off. And if we injected this mouse with a helpful virus expressing HSF1, then the idea is that these mice would hang on and have better stamina. And so we can test that in mice in the lab, literally in the next few months. And that's something that we're doing right now. Okay. So I just want to leave you, I mean, I have a couple more slides, but I just want to stress this point that we can use viruses to protect our muscles. And so I think that there will be a time where we go from, oh no, we have, we've got muscle weakness, we're fighting this disease, to intramuscular injections of a helpful virus that can take us towards a point where we now have either delayed muscle weakness or actually a prolonged period of muscle strength. And we think both of those could be a viable outcome of what we're developing in the lab right now. What about biomarkers? How do we even know who to treat? Sometimes you go to the clinic and they don't even know if you have IBM or something else, right? That's a big problem. So another thing we're doing is we're taking muscle biopsies, and we can use this biopsy needle to isolate a little piece of muscle, and then we can analyze it in the lab. This is a very fancy piece of equipment called a mass spectrometer. And so I just thought I would let you know that we're also identifying biomarkers of IBM. These would be protein biomarkers that might come up in a patient with degenerating muscle fibers. Because if you think about it, we need to know who to treat, right? Sometimes you don't know that you have IBM, but if you do, it'd be better to help stratify which patients to treat. And so that's something else that's happening in the lab right at the moment through the IBM clinic at UNC that we work with. So we have a clinical component at UNC, we have an imaging component, and we have a strong research component. It takes an army, no pun intended to this crowd, it takes an army to do research. And we're fortunate that we have an army of people that can come together with some common interests, but lots of diverse skills. And we can all work together to achieve a common goal, which is to diagnose folks with IBM, image their muscles, and then also come up with new ways to treat them. Of course, this will take, we're in this for the long run. This will certainly take you're in that 5 to 10 year road ahead for the treatment, but we're in it for the long run. And so I just want to end with this idea of healthy living. So some of us have built-in resilience, right? So some of us have this capacity to withstand whatever accumulation of TDP-43 might be coming. We can fight it. And I believe that Uncle Joe, this is my uncle who passed away a few years ago, Uncle Joe had this resilience. He died at 109. This was when
Oh
he
my
was 100.
Lord.
He's 107 at this time. He had just bought a red Mercedes, and he's posing here with his red Mercedes. But Uncle Joe had something that I would call resilience. He could ward off the onset of dementia, IBM, ALS, whatever you think might get a person in their 100s. He had normal vision. He had every vital sign was completely normal. He still walked, he still danced. He was driving up until 107. Remarkable man. And he ended up passing away in his sleep of unknown causes. So nothing that we know of actually caused his death. He was healthy up until the very end. So you might say, why do some of us get ALS, FTD, IBM, and others are resilient up until the very end? And so one thing we're studying in the lab are resiliency factors. What is it in his brain, in his muscle, in his every organ you can name that has caused such resilience? We don't know, but we think HSF1, the thing that I mentioned earlier, might contribute to that. And we're also fishing out other factors that we might find. If we know those, we could potentially both beat a disease, but also create a state of resiliency that makes you, essentially resistant to the pending onset of disease. And that's something that we're really excited about. And this is Joe with his 103-year-old wife who recently passed away as well.
Wow.
So between the two of them, they were 109 and 103, which is really, remarkable. Okay, so this is my last slide. This is our mission. This is my mission, personal mission, but to be done in collaboration with many, many smart and talented and motivated people. Some of them are in the lab, but some of them are patients like yourself. Some of them are at all levels, clinical and basic. We do it together. We do it as a team. If anybody wants to support us, here's my email address and my website. There's really amazing things being done at UNC. I think the culture there is not, there's no egos. So you do this as a group for the right reason to end suffering of patients that have these diseases. And there's no one person that wins. I think we all win. And I think we should be optimistic about what's coming. I also want to let everyone know that if you take diseases from our human history, we've been pretty good at beating diseases when we rally around them. So think about COVID, right? COVID was an anomaly. We rallied around that in a span of, maybe a year and a half, which was remarkable. But even things like HIV/AIDS is essentially, in the US at least, been really almost eradicated. Same of polio, very low levels now. These were once thought to be death sentences. And so we can beat diseases if we rally around them. I think that's the take home message here. If you have resources, creativity, and determination, I think we could really do whatever we put our mind to. So I'll end there. I'm happy to take questions. This is our group again. And these are all the talented people that I work with. And of course, we need funding to make this happen. So I've also listed our funding sources. Some of these come from really amazing foundations, like Muscular Dystrophy Association supports some of our IBM work. and also the Department of Defense, which also supported some of our IBM work. So thanks very much. And Jim, I'm happy to, take any questions from anyone for the remainder of the time.
Dr. Cohen, I cannot express adequately our thanks for your time and expertise. This has been a fascinating presentation, and we are so very grateful that you were able to join us and share this information with us. It is fascinating to listen and learn what advances have been made. There are indeed some questions, one of which is, and you've answered so many of these also in the in your presentation. So, you put that up on the screen. There's a couple of them that were submitted to me earlier that I forwarded to you. So,
Yeah,
you can answer these, please.
Sure, I'm happy to kind of work through the ones you sent me. One was, is there anything I can do to slow the progress of IBM? Yeah, treatments are limited. Many of you may have had steroid treatment, things like prednisone, other treatments that are used with the intention of limiting inflammation. They don't always work. In fact, there are a limited number of patients who have reported that they work well. So I think anti-inflammatories are not going to be the end all. Certainly we need better drugs than those. There have been discussions about immunoglobulin injections. They've worked to varying degrees. The thing I'm most excited about are what's on the horizon, things like immunotherapies and CRISPR-based therapies and gene therapies. So unfortunately, I think most things have not panned out at this point, but I'm pretty optimistic about what's coming. And any clinical trials in the works? There's a couple that I listed here. There's an antibody that might deplete these T cells that are thought to wreak havoc in the muscle fibers. So that's ongoing. There's a phase 2 trial of this drug called sirolimus. That was done in France. It was initially positive, and so that has spawned a later stage trial underway. And there are others, but I don't think they have gained traction yet. So I think IBM is maybe lagging behind diseases like ALS a bit, for which there seem to be many more clinical trials. Maybe all the more reason to put IBM on people's radar. I think it has been viewed typically with less backing, I would say, than ALS. So I think you're all doing the right thing to put this on people's radar. Has there been any research tying exposure of JP4 to IBM? Again, environmental exposures are a lot. Some are hearsay and some are small case reports. There's not a lot of backing of actual solid data for IBM's link to environmental toxins, fuels, things like that. I do believe that's going to end up being true, my personal opinion, but I don't think there's enough data out there to support it, for example. Likelihood of a treatment or a cure in the near future? Boy, I hope so. I think the things that I was excited about during my presentation are the things that maybe have a great chance of working. Things like immunotherapies, things like immune replacement or T cell therapies, gene therapies to deliver genes or pieces of genes that could clear out in this case, TDP43, which is the reason for the degeneration in the fibers. I think those have the most promise. That's just my personal opinion, but you might get a different response for everyone that you ask that question to. The AHR protein, there was a study on this protein, I think from Ben Wolozin's lab at Boston University. We have not worked. Yeah, we have not worked on this protein, but there are toxins that we are screening through from our mega chemical library that are related to this protein that would activate this protein. So We have taken a different approach where we're screening thousands of chemicals in an unbiased way for who causes TDP-43 to aggregate. And these chemicals span every walk of life. And so if a chemical linked to AHR comes out, we'll certainly pursue it. But I was very aware, and I know Ben Willozin, and he did a screen that identified this protein, and it's certainly on our radar. It's very nice work. So thanks for that question.
Yeah, that was something that I had come upon at about the same time I came upon your research, and I had actually contacted Dr. Willozin, and but what was fascinating is that their research showed that dioxins, when injected into mouse tissue, actually increased TDP-43 threefold, and it was a kind of a startling revelation there that had that much of an effect, because many of the dioxins for the veterans that are here in the era in which we served, the dioxins were very prevalent in Southeast Asia, as it was a primary contaminant within the Agent Orange components.
Yeah, and dioxins are in our screen, our current screen. So we're thinking that if they come out and they are a validated hit, we will see them as well. So I'll keep you updated on that.
Yeah.
I want to get to Terry Baker because his hand's been up, and then I'll go through the other questions. Terry,
go ahead.
I got into a program with the VA called the Million Veterans Program. Have you heard of that?
No, I have not.
They wanted, well, I think they've done it now finally, but they wanted 1 million vets to sign up for this. They took, tons and tons of blood. The questionnaire for personal history, family history, environmental history, everything was exhaustive. It was huge. And it's for a big research thing the VA wants to do. And I was wondering if you would be able to access that in any way to get backgrounds or to tie in DNA or whatever.
Yeah, it's interesting, Terry. What was it called? The Million? Can you say
Million
it one more
Veterans
time?
Program.
Million
MVP.
Veterans. Million Veterans Program. I'll look into that. Sometimes the VA has to keep it to within VA, and since I'm not in the VA, it can be some hurdles to jump through. But I'll definitely look into that. Thanks for letting me know that.
Maybe you could even, just get bits and pieces out of there that would be pertinent to you. But still,
right.
It's a really good base if you could tap into it.
For sure.
And they were trying to get veterans from all kinds of different groups, from the really young guys all the way up to us old ones, and have been in Vietnam and the desert and everything else. They were really trying to get this big spread to do a lot of their research.
That's good, because it's hard to get that information otherwise. You really need an exhaustive questionnaire. So if they have that, I'd love to tap into it.
Yes, Todd, this is Dan Butcher. I was Air Force also 29 years, and I just got a request, I guess, within the last week if I would join. I've never joined that yet. I'm not really sure whether I should or not, but it's still ongoing. So
okay.
Join it.
Dr. Cohen, there's some other questions here. Suds, are you with us here? There he is.
Sam.
Suds, did you have questions?
I did. I sent them to you, but like I told you yesterday, I lost my other screen, so I can't see my questions.
Okay, well, I've got your questions. Allow me to relay them if you don't have them readily available. Suds had sent me some questions previously, and the first question is, what is the best way to keep up with the research on IBM being done at UNC?
At UNC, I thought you were going to say in general, with IBM in general, which is what I struggle with.
That too. We'll go ahead and throw that one in as well.
Yeah, I think that UNC is building maybe a more comprehensive website that should allow you to simply search UNC myositis. And I think I'm happy to kind of keep you posted, Jim, as we go forward and
Okay.
relay any new information that'll help you stay up on UNC. IBM in general is a bit of a difficult field at the moment to stay up with. The reason is because of the overlap with ALS and FTD and these other disorders. It's hard to know whether you're reading about one or the other. And then even within the myopathies, there's subtle differences. So I think it's a fair point coming up with ways to stay up. But if you search for IBM, well, SIBM for sporadic IBM and TDP43, if those two keywords go together, I think they'll pull out on a Google search things that are highly relevant to the disease. That would be my best suggestion.
One of the, the majority of the research that I've come upon is concentrated almost universally on ALS.
Mhm.
And is there any kind of for the lack of a better term, of movement, if you will, that would
Yeah.
that would sort of correlate the IBM component more closely to the ALS, to say it's a research for ALS and IBM, because
Mhm.
that will bring IBM into the more into the mainstream, and that's what we need to accomplish as well.
Yeah, and I think you're doing that, Jim. It's advocacy. Part of the problem is ALS is so devastating, and there's a strong emotional connection with patients that have an average lifespan of 3 to 5 years. So ALS is viewed as the disease that we need to fast track. Any therapies, anything that looks promising, ALS is gaining the most momentum. And I think that because it's so devastating, ALS is sort of at the top of clinicians and scientists' lists. But just because you're a rare disease doesn't mean you can't be, that what you've done with moving this up the ranks. And my call with the ALS Association was never so striking as it was on this last call. So the advocacy is probably the best way to go. And a lot of times, yeah.
Forgive me. I was just going to say, can you share with us some of the details of that call? What progress we might be making?
Well, the call was basically putting IBM on the ALS Association's radar. They really knew not much about the etiology of the disease, what's thought to cause it, what population is affected by it. They didn't know much at the highest levels of the ALS Association. Not to fault them, it's just that they have so many diseases that are in their portfolio. They can't study and work on everything. So we basically informed them, myself and Dr. Thomas Lloyd, who was the other letter that was provided.
Right.
We informed them about why it should be on their radar and why it's so intimately connected to the diseases that they study. And it was basically an informational gathering call, but they didn't have much background on it. So it was hey, look, this is the disease. This is why you should care. This is why it's so devastating. This is, we've interacted with, lots of patients that have overlapping symptoms. So it was mostly just an informational gathering call, and we're going to do another call in the future to continue to push the issue. And yeah, and if they recognize it, if someone at that level recognizes it, that's really good news for you all. That will be, it needs to be, yeah.
Go ahead, please.
No, I just think it needs to be pushed. Sometimes there's a political component. Sometimes there's other things that bring things up the ranks, I guess. And in this case, maybe it just took a little nudge from you, Jim, and maybe this ends up really moving up the ladder and gaining traction. And I'll do my best to see it through.
Thank you, because it's something that we know that the two diseases are different diseases. We know that
Mhm.
ALS and IBM are different diseases, but I think what is lacking amongst this community, if you will, is the fact that the disabilities are what we are trying to show people that are nearly identical. The
Mhm.
disabilities themselves, that is what we're trying to convince them of. And I think a number of these clinical types, if you will, are probably, have sort of a tunnel vision from a clinical standpoint, and not the broader actual disabilities that one's face. That's what we're trying to associate. The two diseases are so similar that the disabilities are causing this, the same issues there.
Right.
That's the target we're trying to go after. Now, I wrote
Exactly.
a personal letter to the president and the CEO, and if your continued involvement and the meetings with the ALS are a result of that letter, I am thrilled, and we will.
It absolutely was. The bringing together of myself and Dr. Lloyd and others on a call was a result of your letter. So I think that advocacy, that keeping with it, when they don't respond, you keep sending those letters. I think it really works.
Well, we're
Now,
going to continue, because that is their support is going to be very, very important. Our ultimate goal is to get the disabilities associated with IBM and ALS. Well, ALS already is. We're trying to get the disabilities that are associated with the inclusion body myositis, is to have them recognize this as a service-connected disease, something that,
mhm,
as you said, that veterans are at a higher risk. That's
mhm.
what we're trying to focus on and encourage people to understand.
Yeah, for sure. Well said. I can answer Dick Mason's question real quick. We've screened for chemicals. We've identified 7. These are questions on the chat that I can get through real quick. We actually do them blindly. So we've fished out a category of hits that fall into the agricultural pesticides, things that we're currently spraying crops with. We don't actually know the chemicals and what they are. We just know the family that they fall into. So a lot of the 7, the bulk of the 7, end up being fungicides, herbicides, and other pesticides that are currently being sprayed on apple orchards and other crops and things like that. And so we don't actually know what they are, but once we unblind the study, we'll know exactly what we're dealing with. That's a little scary, right? If you're eating apples or other, vegetables that have been sprayed, and that's influencing. Now, I'm not saying they're causative. I'm saying that they could be risk factors. We don't want to build risk. We want to de-risk. So hopefully that answers your question, Dick. And then David Vok asks, when you say animal models are needed, can you expand on that? It's really difficult to give an animal ALS. A mouse would never get ALS on its own. It would just as soon get eaten by a cat or a hawk or you name it. It's really difficult to give an animal a disease that us humans get. And without spending, too much time on it, building a model that really reflects human sporadic IBM is difficult, and not many people have done it well. There's an investigator at Washington University named Chris Whale. He's done a lot of work in this area, and he's one of the leaders in the field. He's built a model, or maybe 2. We have a model that we're injecting TDP-43 that looks really promising. So the take home here is that make sure you have a model that looks and acts like IBM before you move forward. Otherwise, it won't translate to people.
Could you elaborate a little bit further on exactly what is a model?
Yeah, a model would be something that recapitulates or mirrors the disease in human, but in a different system. Of course, we can't manipulate humans the way we might want to in the lab. So we're forced to use models that we create. And a model would be something where we grow cells in culture, or where we might use a rat or a mouse. Sometimes people use monkeys. The mouse or the rat, a rodent model, has become the model of choice. And when I say model, I just mean a organism that we can use to give IBM to and also to cure it. That's what I mean by model. Thanks for that, Jim. Yeah.
Thank you.
It's been very difficult, and we think our injection model that I showed you might be a good way to give a muscle fiber the TDP-43 that we care about, and then we can go in and cure it. So that would be the idea. Ed Arnold asked, did you mention that you're getting financial support from the DoD? Actually, the DoD funded us several years back to study TDP-43 in muscle. That support has ended. And so we're currently not supported by them, but they helped us out about 5 years ago. And so our support really relies on the government funding, so the NIH, defense funding, DoD, and also philanthropic funding. IBM is not a disease that's on a lot of people's radars, like we just discussed. But when private, either families, wealthy families, other donors want to support IBM research, it can make all the difference, because then you can do things that are high risk, but that a NIH, for example, may not support. You can do things like that with philanthropic dollars. And that's why fundraising efforts are so important. So please spread the word if anybody wants to support this work.
Yeah, we'll pass the hat here.
Okay, yeah, exactly. Marine enlisted either go to Paris Island, South Carolina, or San Diego. That's from Matt. I don't know if that's a question or not, but I guess getting back to areas that might have higher exposures or different, just like you mentioned in Texas. Is there a possibility, this is David, again, is there a possibility that a drug that is effective with ALS may be effective with IBM? That's exactly right. I don't discriminate these diseases. I view them as TDP-43 proteinopathies. That's what we call them. And if a drug works against ALS, the idea would be if you could deliver it to muscle, it should work just the same. And if the diseases are truly due to TDP-43, which we believe to be the case, that's absolutely what we believe. So it could be a situation where when we develop a drug for one thing, it works for these other diseases. That would be amazing. Ken Hampton asked, yes.
No, please continue.
No, I don't want to, I want to make sure I get to your point, Jim.
No, I was just going to ask another question on behalf of one of the other veterans, but if you have
okay.
Please go ahead.
No, go for it, Jim. I was just going to work my way through the list. There's only 2 or 3 others, but why don't you go?
Yeah, this is another question that Suds had requested, and that is, do other universities and professors that are studying TDP-43, do you communicate with these other universities and other scientists other than in publications?
That's a really good question. It really is, because when people are good at what they do, sometimes there's a lack of communication, because they might feel that, well, if I can do it on my own and I can publish my own publications, why would there be this crossover? And just like in any job, there can be egos and there can be people that might want to duke it out to make a discovery, for example. But that's not how therapies and cures happen. You have to work together. You can't reinvent the wheel, right? You can't do all this on your own. So the idea is that we reach out and we become friends and we network with other people. So my philosophy, I can't say this for everyone, but my philosophy is I do my thing well. Why don't we reach out to other people that do their thing well? The issue is IBM doesn't have a dedicated meeting so much. They don't have as many. And so I have to go to ALS meetings in order to run into people that I might network with about IBM. So in the future, it could be the kind of thing where an IBM dedicated venues or conferences, there are some, but I think there's just not a ton of labs doing this type of work. So I think we need to encourage people to do this kind of work, which might get back to advocacy to places like Muscular Dystrophy Association, ALS Association. Put this on researchers' radars. Hey, look, you should be doing this kind of work. And if you want to, here's a call for, sometimes they put out calls for new research opportunities. Maybe there should be an IBM call for scientists doing research. That would be great. So I think we need more people in that field. We need more people to network in IBM, and maybe it can take advantage of the already in place ALS networking, which has really taken off.
Yeah.
So I think that would probably be my suggestion, is to get IBM off the ground with scientists as well. So
That would be helpful indeed, no question about it. The other question the Suds had is do you belong to any other boards as an advisor on any other of these rare disease organizations?
do I belong to any other boards of rare disease organizations? Was that the question, Jim?
Yes, as the ALS or Muscular Dystrophy Association or the ALS Association. And I know that you've been involved previously with the ALS Association if you're not currently ongoing with them.
Right.
But do you contribute to other organizations?
Yeah, I am on so the NIH is the provider of the bulk of the scientific grants that fund people like myself. I am on a standing convening committee for all of the grants that are reviewed by the NIH in the neurodegeneration space. So I do that 3 times a year. And so that takes up the bulk of my time in terms of my obligations for committees.
Okay.
And so I am almost exclusively a member of the NIH and some small foundations here and there. The ALS Association is a new thing for me. But the NIH, since the budget is very high and that supports the bulk of people's work in this space, that's the one that I spend the most time on.
Okay, thank you. Are there there's a question from Augie. Augie, go ahead. You got to unmute yourself there, please.
Okay, great. Dr. Cohen, I wanted to get your take on something. I was recently approved for service connection for my IBM. And in the approval letter, they, because in my evidence that I submitted was my exposure while I was in the Navy to a variety of toxins, toxic environmental factors, including ethylene glycol, which was de-icing fluid used on the aircraft that I flew. And interestingly, in the letter that I received from the VA, they specifically made mention to my exposure to ethylene glycol, and they stated that that has been associated with the onset of ALS. Are you aware of that actually? That was the first time I've heard of any kind of an association of anything specifically to the onset of ALS. Are you aware of that?
I have not been aware of that. Some of these are a little bit hearsay, and I just want to take those comments with a grain of salt, because there could be a very small study here or there that someone might use as grounds for that. I'm not aware of that, but I'm happy to look at if they provided you with a paper or some information that might help me understand it, I'm happy to take a look at it. From what I can tell, the link between these things and ALS, the one I mentioned in Guam, the ALS of Guam, that has the most data to back it. Everything else has been a little bit hearsay and a little bit sort of, we think it's possible, potential, those kind of words. But if they provided you with any information, Augie, please send it to me and I'm happy to look it over if that's useful.
They did not provide any information. The only thing it was just a one sentence listed in my approval letter. But the other thing that I would like to point out is that while ALS itself is so devastating because of the short timeline, which I'm guessing is that because it's starting in the brain and it's got more direct access to the nervous system. But I'd also like to point out that having to look at the last 10 to 12 years of your life in a totally disabled state is equally as emotionally disturbing. It just takes us longer to get there and we have more time to adapt along the way. But, you know, like Jim had pointed out, the disabilities associated with each of those diseases, SIBM and ALS, are so similar that this disease should be considered every bit as debilitating as ALS. And thank you for your participation today. I really, really appreciate it.
Thank you. Good point, Augie. Appreciate that.
We are getting close to our time limit here. Are there any last questions that Dr. Cohen can field for us? If not, then Dr. Cohen, I cannot thank you enough for sharing this time. But this has been fascinating. And I think anybody who is here listening to your presentation has gained a lot of information that was previously unknown. And I just am so very grateful that you were able to spend this time with us and give us this information. If you could continue to contact me on any of these other slides and things like that, then I can disseminate them to the rest of the members here. And we will continue the fight. We will keep in contact with you. And we are very interested in knowing what the ongoing dialogue is with the ALS Association, along with you and Dr. Lloyd, because that's going to be helpful. With that said, I will let you know that we have a letter that is going out to both the House Subcommittee on Veterans Affairs and the Senate Committee on Veterans Affairs that contain your letter. And I think that is going to be very influential in getting veterans recognized or getting IBM recognized as a service-connected disability for veterans. And so for that, we are eternally grateful for that help.
Absolutely.
And
the pleasure goes both ways. I really enjoyed all of your questions, and I wish you the best as you get treatment and start, finding ways to navigate the debility. I hope that whatever we can do in terms of this letter helps as we try to fight for, more rights, which I think you all very much deserve. So
thank you so very much. And with
Yep.
with the topic of that letter, to all of you who have responded to my requests for your signatures and your military data, we have 40 signatures that are accompanying that letter. And I am so very grateful for each and every one of you that has shared those your signatures that we put on this letter. For those of you who have not sent your signatures in, please consider doing so. The more signatures that we have on this letter, the more impactful it is going to be. And when you see a letter asking Congress to change the laws on our benefit, and we back it up with the signatures of 50 disabled veterans from IBM, it is going to be meaningful. So if you have not sent a signature in to me, please do so. All you got to do is sign just a white piece of paper, scan it to a PDF file, and include it into an email. Otherwise, put it in an envelope and mail it to me, and I'll attach it to that letter. So to all of you who have helped with that, we thank you again. Dr. Cohen, thank you so very much. We're so very grateful for your help.
Jim, can I put a quick plug in for our next month program?
Oh, by all means. Yes, Augie, thank you. Go ahead.
Yes, yeah. Next month, make sure that you come back, because we were going to have the attorney that I used and Larry Lesher used to gain our approvals for service connection for IBM. He is going to be with us next month to talk about the process and ways to enhance your likelihood of getting an approval letter from the VA. So make sure that you're here September 9th, and we can pick his brain.
Indeed. Thank you, Augie.
One other thing, Jim, for Todd, I'm sending my wife Nancy out to Piggly Wiggly to pick up a bucket of resiliency for us up here for this afternoon, and telling her don't get those bats anymore, because we're not going to be eating those.
Yeah, I was going to tell you, if you have raw bats on your plans for dinner tonight, please consider drive-throughs or carry-outs.
If Piggly Wiggly sells raw bats, I'd be pretty impressed.
Okay.
All
To
right.
everyone, thank you for being here, Dr. Cohen, again.
All right. Thank you, everyone.
Endless thanks. We will say goodbye, and thank you to all.
All
Thank
right.
you.
Take care. Bye-bye.
Thank you.
Bye.
Bye.
Thanks, Joe.
Thanks, Jim.